Welcome, everyone. Let me introduce the topic of my PhD research, which is investigating the role of probiotics in shaping the human gut microbiome. My name is Éliás, and I am a dietitian for profession and working currently at the Faculty of Health Sciences at Semmelweis University. My aim is to formulate evidence-based and applicable recommendations for the use of probiotics in several conditions. Therefore, my mission is to understand the role of the human gut microbiome in health and disease and the consequences of treatments on the gut microbiome. I have two currently running projects. First, I investigate the effect of probiotics for the prevention of antibiotic-associated dysbiosis. And after that, the second topic is investigating the effect of probiotic supplementation in the basically healthy population. So the first question is, do probiotics prevent the dysbiosis of the microbiome during antibiotic therapy? Here you can see the status of this project. We are currently in the status of article writing. We will finish it soon with my supervisors, so hopefully we can send it to the internal review next week. And our target journal for publication is the Gut Microbes. A little bit about the background. So probiotic supplementary market is $1 billion one, which is still expected to be growing. Probiotics are used in several conditions, but probiotic supplementation is 9 times more likely during antibiotic therapy. This is because the antibiotic-induced dysbiosis is a potentially harmful state with the overgrowths of harmful bacteria, which can lead to, for example, acute diarrheal disease or Clostridium difficile infection. However, the role of probiotics in preventing this dysbiotic state is still in question, and actually there is a knowledge gap in the guideline as well. Therefore, our aim was to investigate the effect of probiotics for this antibiotic-induced dysbiosis. So our population, which we investigated, was patients treated with antibiotics. The intervention is the use of probiotics simultaneously, and the comparison is no probiotics or placebo, and the outcome is the gut microbial diversity and composition. Here you can see the workflow of the systematic search and selection. At the end, we were able to include 15 articles in the systematic review part and only 4 in the meta-analysis. Here you can see the results of the investigation of Shannon Diversity Index, which is one index that can describe diversity of microbiome. Here you can see that patients who were simultaneously supplemented with probiotics during antibiotic therapy had a significantly higher diversity index at the end of the treatment. And you may notice that there is an outlier article, the first one. In this case, they investigated healthy population without any medical reason for antibiotic therapy. So therefore, we also performed a subgroup analysis without this article, and here you can see that the effect is even more pronounced. However, I would highlight that in the systematic review part, we had much more outcome because there are many indices that are used in the literature describing the diversity. And it varies among the articles which is reported and which is not. For example, regarding the Shannon Diversity Index, you can see that we have more articles that reported on that, but we were able only to analyze quantitatively 4 of them, because the other articles only use qualitative reporting, so just mentioned that there was no significant difference. So it's a confusing factor regarding the result. And if you see, the black cells shows no significant difference between the group, which is the majority of the cases. The similar tendency we can observe regarding beta diversity indices. The before mentioned table was describing the alpha diversity indices. So the main strength of this study is that it's the first meta-analysis in the topic, and it represents the highest level of evidence because we only included randomized controlled trials. However, we have some limitations because we were able to analyze only low number of studies with low sample sizes, and sometimes we had to use other methods for the transformation of data from box plots because the data was not reported quantitatively. Since we were able only to meta-analyze one index of diversity, it is not really sufficient according to the literature because you could see that there are many ways to describe this diversity. And the studies that were included in the analysis were very heterogeneous regarding the duration, the type of antibiotics, and also the reason of the antibiotic therapy, and also the probiotic strains were different. In conclusion, we can say that the meta-analysis of Shannon Diversity Index showed a significant effect of probiotics on the diversity. However, as I mentioned, we were not able to quantitatively analyze all the data that we have, which is a confusing factor. And the thing that we could only analyze this one index, it is not really appropriate way to say that we can promote the probiotic supplementation to maintain diversity, because the summarized results of the systematic review are not convincing enough in this regard. We also try to summarize the results of the taxonomic composition, which I could not show here. But in conclusion, I can say that the changes are similar in both groups, regardless of probiotic supplementation. And we can also see that there is a tendency of the restoration of the microbiome in 3 to 8 weeks of follow-up in both groups. So at the end of these follow-up periods, also the differences between the two groups disappeared. So we can say that probiotics tend to have only a limited and temporary effect on the gut microbiome, so the usefulness is questionable during antibiotic therapy, but at least we can say that they are probably not harmful. The implication for research will be that we see that there is a lack of standard units of measurements and no standardized follow-up periods and study protocols. If we would have these things, the results would be more comparable and valuable for us. And we also need clear cutoff values for ideal microbiome diversity indices. The second project of mine is investigating the effect of probiotic supplementation on the gut microbiome composition and also other clinical outcomes in the healthy population. It is important because nowadays we can see the trend that also healthy people take probiotics without any medical reason. However, we don't know the effect of this supplementation on the health and on the microbiome in this healthy population. So we really need consistent and practical recommendations. The investigated population will be the general healthy population, and we will compare the taking probiotics and not taking probiotics, and we are going to investigate like any outcome that is reported in the articles, like cognitive function or gastrointestinal function and everything that is in the literature. And according to our hypothesis, the probiotic supplementation will not alter the composition of the gut flora in the healthy population, but may have an impact on other health outcomes. Here you can see the search key for this project, which we made the systematic search in March. And we are currently in the phase of title and abstract selection. So I would like to submit the first paper in June and the second in September. Thank you for your kind attention, and I am happy to answer any question that may arise. Thank you.
As we know, there are a lot of methodological approaches to define the microbiome from the stool, like rRNA-based methods and shotgun methods. Did you also consider this problem, or even the algorithms are different, pipelines are used, and maybe these also influence the results of the different studies? What do you think about this?
Yes, thank you for pointing that out. Yes, there are several ways to investigate the microbiome. The included studies use this 16S rRNA technique. The concrete algorithm I don't know because they didn't report it, but yes, so all the studies that were reporting on diversity indices used that technique. And there were some other articles that were included in the systematic review part where we analyzed or summarized qualitatively the results of the taxonomic composition. So there were some articles that use standard microbiological methods, which is obviously a whole other thing. But of course, we have it in mind, and it is written in the article, so yes.
Thank you for your presentation. My question is regarding to your second project. Is there any dietetic restrictions in these studies, or do you expect to have? What do you see?
You mean like for the inclusion criteria? Yeah. Yes. Usually they only asked, I mean, the patients that are patients, okay, they will be healthy population, but okay, let's call them patients. So they are usually asked just not to take probiotics, but not, so like probiotic food, like yogurt, I think you, yes, you wanted to ask this. In most of the cases, this is not restricted, actually. So yes, this is a factor that we have to consider.
What would be the significance, or what would you think about changing the so-called healthy people or populations gut microbiome? What would be the benefit of it, or why do you assume that anything would change it, which is relatively stable after reaching adulthood and until late elderly age? It is quite stable in under healthy circumstances.
Yes, thank you for the question. So I think there is no reason to take antibiotics when you are healthy. So that's why actually I chose this topic to show that it is unuseful, yes, to take. So there are many people that take it, but like, why? So don't spend money on something that is...
So you don't expect any change?
No, I don't expect.
Okay, I missed that. So in that case...
Yes, so that was the hypothesis, actually, that we don't expect any changes, yes.